Hi everyone, in this video we will discuss few questions from fatty acid metabolism. So the first question is Carnitine shuttle. It is a 3-mark question. So we should know regarding beta oxidation of long chain fatty acid that takes place within the mitochondria. So prior to this beta oxidation, that fatty acid has to be activated. That activation reaction takes place within the cytoplasm. So after this activation, this long chain fatty acid has to be transported to the mitochondria for the oxidation reaction. But the problem is regarding the inner mitochondrial membrane. That inner mitochondrial membrane is not permeable for the long chain fatty acid. So this long chain fatty acid require a transporter molecule to transport through the inner mitochondrial membrane, and that transporter molecule is known as carnitine. So carnitine is a carrier molecule to transport long chain fatty acid across the inner mitochondrial membrane. That long chain fatty acid is very important because short chain and medium chain fatty acid does not require this carnitine molecule. So in carnitine shuttle, you have to mention about these 3 enzymes. One is carnitine acyl transferase 1. That enzyme reaction is to produce acyl carnitine, and the second enzyme is translocase, and that enzyme action is to transport this acyl carnitine across the inner mitochondrial membrane. And the third enzyme is carnitine acyl transferase 2. That enzyme mediates the release of acyl group from the acyl carnitine. What are the amino acid composition of carnitine? So carnitine is a protein molecule, so it is made of amino acids. So the amino acid compositions are lysine and methionine, and the synthesis of carnitine takes place within the liver cell. And the second thing is, we should know the chemical name of this carnitine. The chemical name of carnitine is beta hydroxy gamma trimethyl ammonium butyrate. That is also important thing. Chemical name and the amino acid composition. The amino acid compositions are methionine and lysine, and the chemical name is beta hydroxy gamma trimethyl ammonium butyrate. And the next question is carnitine deficiency. It's a 2-mark question. So carnitine is required for the oxidation of long chain fatty acid. So its deficiency leads to defective beta oxidation of long chain fatty acid. Then you should know what are the clinical features of carnitine deficiency. The clinical features are, one is fasting hypoglycemia, and it will produce as muscular weakness. So why there is a fasting hypoglycemia in carnitine deficiency? So during fasting state, the blood glucose level is maintained by gluconeogenesis, and this gluconeogenesis is a energy dependent reaction. This reaction requires ATP, and this ATP is contributed by beta oxidation during fasting stage. So in case of carnitine deficiency, there will be defective beta oxidation. So that will lead to defective gluconeogenesis. That defective gluconeogenesis is responsible for fasting hypoglycemia. And here the treatment is oral supplementation of carnitine. And next question is biochemical defect in Refsum disease. It's a 1-mark question. Similar to beta oxidation, there is another oxidation that is known as alpha oxidation, and alpha oxidation is important for phytanic acid. Phytanic acid is an important fatty acid present in breast milk. So phytanic acid undergo alpha oxidation, and it is mediated by an enzyme known as alpha oxidase. So deficiency of this alpha oxidase enzyme resulting defective alpha oxidation of phytanic acid. That is the biochemical defect in Refsum disease. So defective alpha oxidation of phytanic acid, that is the biochemical defect in Refsum disease. And the next one is a essay question. Name the site where beta oxidation of fatty acid occurs, describe the steps, and explain how much energy is released during the beta oxidation of one molecule of palmitic acid. So here you should write the site of beta oxidation, that is the mitochondria of the cell. And the starting material is fatty acid, and the end products are acetyl-CoA, and it also produces NADH and FADH2. Then you should write the steps in beta oxidation. So the first step is the activation of fatty acid that takes place within the cytoplasm. Then you should write the role of carnitine, and next you should write the beta oxidation steps, that is oxidation, hydration, again oxidation, and finally there is a cleavage step. So first oxidation, it is mediated by FAD dependent dehydrogenase enzyme. So the first oxidation produces one molecule of FADH2, and followed by one hydration reaction. Then it is again second oxidation, that is mediated by NAD dependent dehydrogenase, and this reaction produces one molecule of NADH. So beta oxidation, in each cycle it produces one molecule of FADH2 and one molecule of NADH, and this FADH2 and NADH enters into the electron transport chain where it produces ATP. So regarding the energetics in beta oxidation of palmitic acid. So palmitic acid contains 16 carbon atoms. So it can undergo 7 cycles of beta oxidation. So each cycle of beta oxidation produces 1 molecule of FADH2 and 1 molecule of NADH. So 7 cycle produces 7 FADH2 and 7 NADH2. 7 FADH2 produces 10.5 ATP and 7 NADH produces 17.5 ATP. And finally it produces 8 acetyl-CoA because it is a 16 carbon compound. So 16 divided by 2, it is 8 acetyl-CoA. Acetyl-CoA is a 2 carbon compound, and each acetyl-CoA enters into the TCA cycle. In TCA cycle, each acetyl-CoA produces 10 ATP. So 8 acetyl-CoA produces 80 ATP. So in total, this beta oxidation produces ATP. But the initial activation reaction requires 2 ATP. So the net ATP yield in beta oxidation of palmitic acid is 106 ATP. That is the total energetics in beta oxidation of palmitic acid. So that's all regarding the metabolism of fatty acid. Thank you.